Hello, everyone. Hello. Bonjour, Europe. Good morning, Europe. Guten Tag, Germany. Dobar dan, Serbia. Dobry dzień, Russia. Do we have some
Konnichiwa.
people from... Shima-san! Hello! Oh, I'm so happy.
Elena-san, como ça va?
Hi, Antonella.
I'm very, very good.
How's it going, Mania?
I'm so happy to
George.
see
Hi, George.
our new friends
George.
from
Hi, good
Suiza.
morning.
Good morning.
Hello, Tomoko-san.
Yes.
Oh, Uchida-sensei. Hello. Miriam. Wow, so many nice people. Ah, konnichiwa. Actually, konbanwa, Nihon. Konbanwa. Yoroshiku onegai iteshimasu.
Konbanwa.
Hi,
Okay.
Mayumi-san.
Hi.
It is
Konbanwa.
5:01 and we can begin our November webinar today. And for a start, I would just like to ask everyone, if possible, could you please, all of you, turn on your videos so we can see each other and our speaker can see actually his audience. It is always much better feeling when we see each other and we know to whom
Thank
we are
you.
talking to. Hello. So today we are having a very, very interesting lecture, and we are both grateful, Professor Tenkova and I, to Joachim Keppler, who kindly accepted to give this talk today, which is a very nice continuation of the lecture that we listened on our previous webinar with Professor Giuseppe Vitiello. So today we are again going to listen about water and consciousness and brain. So for those of you who do not know Professor Keppler, he's a physicist and his main actually topic of research is a fundamental mechanism of consciousness, which I think is one of the most interesting topics a scientist can actually explore. So before we begin, maybe Professor Tenkova would like to say something about Professor Keppler.
Yes, thank you.
And
We know each other since his recent papers, and we met at the conference, and we enjoyed very much our discussion. And thank you very much, Joachim, for agreeing to share with us your recent studies. We are on the line about brain and consciousness recently, and coherence, starting with Antonella De Nino and then Giuseppe Vitiello. So we will continue going back and forward to aquaphotomics and trying to see the connection between water and consciousness and brain, and what actually aquaphotomics, how aquaphotomics can be a powerful tool in this direction. I'm looking forward really to your talk. Thank you very much again. Good luck.
okay, thank you. So just before we begin, I would like to add, I do not know much about Professor Keppler's work. I just recently, maybe like 6 months ago, heard about one of your papers that was presented to me by our lab member Nishiyama-san from Tanaka-sensei's laboratory. And it was a very, very tough webinar that I attended because the paper was presented in Japanese. But despite that, despite listening in Japanese, I was so fascinated and I loved what I heard. So you have my interest. And now you can, I think, begin. You have 30 to 40 minutes, and afterwards we will have a discussion. If anyone in the audience has questions, please type them in the chat box, and afterwards, of course, you can join live. Yes, Professor?
No, I just I'm saying hello to people that are coming in. Yes.
Joachim, if you're
Sorry.
ready,
Yep.
you can begin. Thank you so much.
Thank you very much. I first of all share my screen. So
Okay, we see. Very nice.
we just wait a minute. this one. Yeah. So let me do it like this. So, well, now you should see my screen.
Yes.
And first of all, thank you very much for the invitation and for the introduction. Yeah, well, good evening from Germany. It's actually, it's morning in Central Europe, but it's evening in Asia. My name is Joachim Keppler, representing the Davis Research Institute. And welcome to the seminar, which is about consciousness, more specifically about the role of the brain's neurotransmitter-water matrix in the formation of conscious states. The agenda of the talk is as follows. I would like to start with some thoughts on how to link consciousness to the foundations of physics and how to reconcile consciousness with the laws of nature. Then I would like to turn to the functioning of the brain and the role of the neurotransmitter-water matrix in the processes that take place in the brain. And finally, on the basis of these findings, on the basis of our discussion in part 2, I will address the mechanism underlying the formation of conscious states, which I hope will lead us finally to a self-consistent theory of consciousness. So let's start with some thoughts on how to link consciousness to the foundations of physics. For this purpose, I would first of all like to set the scene and clarify what consciousness actually is. Well, this is me, I'm a human being, and just like all of you in the audience, we are sentient beings who are endowed with an inner experiential world, which means that we experience conscious states. And the defining characteristic of conscious states are phenomenal qualities, so-called qualia, meaning that a state of consciousness is always distinguished by the presence of qualia, which could, for example, be the taste of a strawberry or the experience of a colorful sunset or just a moment of happiness that I had when this picture was taken. So these are all phenomenal conscious states, and we want to get an understanding of how a material, a physical system can give rise to consciousness. Now, one of the most challenging questions is how consciousness, how phenomenal qualities can be reconciled with the laws of physics. The great achievement of physics is to have deciphered the construction plan, or most of the construction plan of the universe. So what you see here is a very compact mathematical formulation of the so-called Lagrangian of the standard model of physics, which describes, on the one hand, the fundamental building blocks of matter. These are the particles, the elementary particles. And on the other hand, the fundamental forces, which are represented by ubiquitous energy fields. And of course, there is also a dynamical interaction between the fundamental constituents of matter, the elementary particles, and these fundamental force fields, which is also encapsulated in this mathematical formulation. And from a physicist's point of view, it seems natural to develop a theory of consciousness that manages to link consciousness to the foundations of physics and integrates qualia at the end of the day seamlessly into the scientific worldview. And when you actually look at the field of consciousness research and follow this field, you see that there are actually, or currently, different lines of approach to achieving this goal, with each route encountering its specific challenges. Before I come to my approach, I would like to say a few words about the route that is still being followed by the majority of scientists, mostly also by the neuroscientists. And this route is known as conventional physicalism. And the proponents of conventional physicalism fully accept the achievements of physics. They believe that there is, well, this fundamental understanding of nature, the physical worldview, but they believe at the same time that there is no consciousness whatsoever at this fundamental description level of nature. And instead they hold the view that the emergence of consciousness is linked to the exceedance of a critical level of complexity, which means that qualia, in their view, from this conventional physicalism view, that qualia are believed to be the result of complex processes or complex computations taking place in the brain. And, well, this at first sight sounds plausible, but on closer inspection, this approach is afflicted with many, many serious explanatory gaps. I cannot go into the details here. I just want to summarize the main problems. And these are, at the end of the day, it's a lack of a plausible mechanism for the generation of phenomenal properties from insentient matter. You simply cannot understand why phenomenal properties should emerge from insentient matter. And this is because there is no generation mechanism that is intelligible. Yeah? There is no comprehensible functional principle of a conscious system based on this view. And therefore, we also do not have tangible demarcation or tangible threshold criteria for conscious systems. So in summary, this approach has neither, when you look closely at this approach, it has neither explanatory nor predictive power, as it cannot tell us what level of complexity is to be considered sufficiently high or sufficiently complex to transcend the threshold of consciousness. And this means that what I'm calling here strategy A or line A, namely the view that consciousness is not inherent in the construction plan of the universe, that consciousness is a cosmic latecomer, that this strategy, that this approach is not very promising. Yeah? It has severe explanatory gaps, and there is, for me at least, no progress visible following this path. In understanding consciousness, in understanding phenomenal qualities. There is a second line of approach, which I call here line B, and this is based on the assumption that consciousness is inherent in the building blocks of matter. And if you follow this line, you are a so-called micro-level panpsychist. And unfortunately, you will sooner or later also arrive at severe explanatory gaps, the most prominent of which is a so-called combination problem. I will also not go into the details here. My personal take is that because of these problems, this approach will also not lead us to a robust theory of consciousness. The line of approach I am following, and which I would now like to focus on, is strategy C, it's line C here, which assumes that consciousness is inherent in the ubiquitous energy field. And for this novel approach, the term cosmopsychism was coined. And as we will see, I consider this approach to be the most promising, as it circumvents the explanatory gaps of all the other approaches. And the basic idea behind this novel approach is to place special emphasis on the omnipresent energy fields that are included in the construction plan of the universe. And these energy fields are subsumed under the umbrella term zero-point field, which has several constituents, so to speak, the electromagnetic part, the Higgs part, if you like, the weak and the strong interaction part. But the electromagnetic part will be the one that we will focus on later, in the discussion. But just to give you an impression, all these fields, these ubiquitous energy fields that take a prominent role, or play a prominent role in the cosmic blueprint, they are somehow subsumed under this umbrella term zero-point field. And this field can be decomposed into a spectrum of normal modes, which means that in this intrinsic or extrinsic physical manifestation, this omnipresent field is believed to be, or it can be described as a spectrum of frequencies, so to speak. It consists of the full frequency spectrum, and you can think of this omnipresent field as being decomposed into a frequency spectrum. And from this point of view, it's also something like a formless and undifferentiated ocean of energy that pervades the whole cosmos. And what I'm telling you here is all well-known physics. It's fundamental physics, and these building blocks of matter, these energy fields, can be described clearly in physical terms. Now I do one step, namely one single postulate. I postulate that this field is actually a dual aspect entity with an extrinsic physical manifestation, but also an intrinsic phenomenal essence. So the postulate is that this field is not only a physical, but also a phenomenal field, and that each normal mode of the field is not only associated with a specific frequency, but also with a specific phenomenal quality. So activating a specific frequency means activating a specific phenomenal quality. So that we are not only dealing here with a formless, undifferentiated ocean of energy, but at the same time with a formless, undifferentiated ocean of consciousness. And at the end of the talk, we will see that this approach will open up completely new perspectives for our understanding of consciousness and will remove the obstacles on a way to a theory of consciousness. However, before we get to that point, I would like to make a cut. And I think we first need to understand the functioning of the brain before we can appreciate the role of the zero-point field. That's why in the second part, I would now like to come to the functioning of the brain and the role of the neurotransmitter-water matrix. To this end, we first of all take a look at the architecture of the cortex. So the cortex is organized horizontally in layers and vertically in columns. And it's widely accepted that the basic unit of operation of the cortex is a so-called microcolumn. A typical microcolumn consists of roughly 80 to 100 neurons, most of which are so-called pyramidal neurons that are equipped with branched dendritic trees. And these dendritic trees are covered with synapses through which the neurons receive their input signals. And each synapse is, again, a very complicated object. It itself consists of a so-called presynaptic terminal that contains vesicles with a high concentration of neurotransmitter molecules, the release of which leads to the activation of receptors located on the postsynaptic terminals. And so what you see here is it's essentially the hierarchical organization of the cortex. And the key message on this slide that I would like to convey is that an important finding of neuroscience is that the cortical surface consists of a vast number of these microcolumns with little variation in size and a typical diameter of 30 micrometers. So this is the essential functional unit of the cortex that plays a major role in all cortical dynamical processes. And we are talking here about this unit, this cortical microcolumn with this size of 30 micrometers. Now let's take a closer look at the structure of a cortical microcolumn. A microcolumn receives numerous inputs from other cortical areas and also the thalamus, and these so-called cortical and thalamocortical fibers connect to the synapses that populate the dendritic trees of the pyramidal neurons. And in active synapses, signal transduction takes place through the release of neurotransmitters, which is mainly excitatory through glutamate. And the glutamate pool is regulated by glial cells, the so-called astrocytes. And finally, we also have an output, of course, of a microcolumn, which goes through axonal outputs. And through axons, at the end of the day, signals are transmitted to other regions of the cortex. So this is basically the picture, and it looks rather complicated, but when you look at the picture from a physicist's point of view, it can be translated into a simplified model of a microcolumn that includes only pyramidal neurons and glutamate as an excitatory neurotransmitter. To be precise, the model relies actually on two glutamate concentrations, namely on a peak concentration encountered in the synaptic vesicles and an average tissue concentration, which is regulated by the cells on the left-hand side, these astrocytes. So in summary, from a physicist's point of view, a microcolumn can be regarded as a bundle of pyramidal neurons encased in a glutamate pool, or more precisely, in a glutamate water matrix. I think it's important to note that glutamate and water are actually the most abundant molecules in neural tissue, and they, via hydrogen bonds, they form a glutamate water matrix. So, in summary, the brain consists of a vast number of these microcolumns, these basic functional units of the cortex, and each unit consists of a bunch of pyramidal neurons that are surrounded by a glutamate water matrix. That's a simplified picture behind the brain. It's a vast array of these microcolumns, and each microcolumn is, in simplified terms, nothing else but a bunch of neurons that is embedded in a glutamate water matrix. The next step is to arrive at a field theoretical description of a microcolumn. For this purpose, we need to know that according to quantum electrodynamics, which is the fundamental theory of the electromagnetic interaction, the vacuum is everything else but a void. It's permeated by omnipresent fluctuations of the electromagnetic field. And these fluctuations are referred to as a zero-point field. So this is now the point where we revisit the zero-point field. And as we've already seen before, the zero-point field can be decomposed into a spectrum of normal modes, which are the elementary frequency components of the field. And according to QED, this fundamental theory of the electromagnetic interaction, it is this field, this omnipresent background field, the zero-point field, that can be seen as the ultimate basis, the fundamental substrate of the electromagnetic interaction. Now, one can achieve a QED-based description of a microcolumn, which takes into account the resonant interaction of the zero-point field with the glutamate water matrix of a microcolumn. And such a description results in equations expressing the dynamical evolution of this coupled glutamate zero-point field system. And the key finding is now that upon exceeding a critical coupling strength, a phase transition is triggered and the whole glutamate pool of a microcolumn is driven toward a coherent state. And this criticality, this threshold criterion is tied to the concentration of the glutamate molecules and their vibrational excitations that are amplified in the glutamate water matrix. So we have this glutamate pool, and this pool interacts resonantly with the zero-point field and gives rise to the formation, to the emergence of a coherent state. And the basic criterion for all this is the concentration of the glutamate molecules, and water plays the role of an amplifier in this matrix. Water amplifies the resonant behavior of the glutamate pool that is coupled to the zero-point field. That's the story behind now the zero-point field interacting with this glutamate water matrix that is a basic part of the cortical microcolumns. Based on these insights, we are now in a position, well, to formulate or develop a functional model of a microcolumn. So what does it do now? What does a microcolumn actually do? And in this model, the synaptic vesicles, where we find the peak concentration of glutamate, they play the role of the individual clusters which trigger a phase transition by resonant glutamate zero-point field interaction. So if many synapses are activated simultaneously, a phase transition is initiated that pervades the entire microcolumn. And now there are two important consequences of this resonant glutamate zero-point field interaction. First, it gives rise to the emergence of a collective state in which all the molecules behave collectively. They reside in a superposition of a ground state and a vibrational excitation. And most importantly, the amplitude of the resonant field modes, those field modes that play a crucial role in this resonant interaction, they are significantly boosted. They are amplified. And this is what we call the runaway stage. And the runaway stage sets off an avalanche process that drives the evolution of the entire glutamate pool toward a stationary coherent state. And this state is then, or the dynamical properties of this state are determined by the tissue concentration of glutamate. So in summary, the functional principle of these basic units of the cortex, these cortical microcolumns, relies on resonant glutamate zero-point field interaction, which results in the formation of a coherence domain and leads to the amplification of field modes of the zero-point field. So this is how a microcolumn works. And this functional model is not only a nice idea, it's based on a rigorous mathematical analysis that demonstrates the viability of this model and shows that these processes are really feasible under the conditions encountered in the brain. Yeah? So the story here I would like to, or the message I would like to convey is that all this, what I'm telling you, it's not only a nice idea. a pure hypothesis. It's actually based on rigorous mathematical calculations, on quantitative calculations. And in order to demonstrate the viability of the model, one has to perform calculations. However, I just want to briefly go in here. I do not want to go into the details. I don't want to bother you with the details. I just want to mention that what I'm telling you here is part of a rigorous mathematical analysis. For instance, just to give you an impression, one question would be, well, given the peak concentration of glutamate in vesicles, is this concentration really sufficient to initiate the runaway stage? And doing the calculations and so on, you see, well, the answer is yes, indeed, the coupling strength lies in the critical regime. A second question, for instance, would be, well, does resonant glutamate zero-point field interaction really result in a stationary coherent state? And the answer is again yes. The calculation shows clearly that a stationary solution can be found, and you have a specific resonance frequency that drives this evolution of the glutamate pool and leads to a stationary state. So altogether, it's 5 critical questions that need to be answered, and the message I would like to convey: these answers can be given, and you find that the model is viable. So instead of going into the depth of the calculations, as I mentioned, I would like to present the conclusions that can now be drawn from these mathematical calculations. First, the quantitative calculations demonstrate that macroscopic quantum coherence in the brain is feasible. Macroscopic quantum effects are feasible. This can be shown mathematically. More specifically, the model calculations now suggest that the functional principle of microcolumns, of these basic units of the cortex, relies on the coupling of the zero-point field to the glutamate water matrix. So following these calculations, the clear suggestion now is that the brain seems to be specifically designed to modify the zero-point field. So that's actually obviously the role or the function of these units, namely the amplification of selected zero-point field modes. So the picture now takes shape that long-range synchronization in the brain emerges through a kind of bottom-up orchestration process involving the zero-point field. And the basic characteristic of this process lies in the formation, propagation, and synchronization of coherence domains. So in simplified terms, the picture looks like this. A stimulus gives rise to the activation of selected microcolumns. Each of the activated microcolumns forms a coherence domain. And the formation of coherence domains leads to downstream effects that are crucial for long-range synchronization. And why I'm telling you this, the important point here is that long-range synchronization is a key characteristic of the neural correlates of consciousness, meaning that we are now able to explain the processes behind the neural correlates of consciousness. So the main insight from the model is that the whole process is based on resonant coupling of the zero-point field to the glutamate water matrix. And this process results in the amplification of selected modes of the zero-point field. And it is this insight into the mechanism that takes place in the brain that now opens up completely new perspectives for the development of a theory of consciousness. The central idea being that the key to the understanding of consciousness lies in the zero-point field. It's not the brain, it's just the neural correlates of consciousness, the dynamical characteristics of the brain that can now be described, but you still cannot understand the phenomenal properties of consciousness. And the key to understanding now phenomenality and consciousness itself, obviously, or a hint is now that this may lie in the zero-point field. So what conclusions can be drawn about the mechanism underlying the formation of conscious states, and what can the path to a theory of consciousness look like? For this purpose, we now return to our core hypothesis, which regards the zero-point field as a fundamental psychophysical backbone of the universe. In this section before, in the second part of the talk, in the previous section, in this field theoretical model that I explained to you, we have been dealing with the extrinsic physical manifestation of the zero-point field. So once again, this field consists of a spectrum of normal modes. It can be regarded as a formless and undifferentiated ocean of energy. But now the hypothesis is that the zero-point field has not only this extrinsic physical manifestation, but also an intrinsic phenomenal essence, which means that each normal mode is also associated with a specific phenomenal quality, and that we are dealing here with a formless ocean of consciousness. Now, proceeding from this dual aspect picture, let's now look at the resonant interaction between the brain and the zero-point field. From the physical perspective, resonant brain zero-point field coupling, which is, as we have seen, resonant coupling of the zero-point field to the glutamate water matrix. This results in a set of amplified normal modes of the zero-point field. But now, from the phenomenal, from the intrinsic perspective, this can be interpreted as an amplification and activation of a set of phenomenal qualities inherent in the zero-point field, resulting in a unified state of consciousness that emerges from the background of undifferentiated consciousness. So in a way, in this understanding now, the brain produces concrete conscious states by playing chords on the keyboard of the zero-point field. And the distinguishing feature of this process should be the formation of coherent states, which manifests itself in synchronized large-scale activity patterns in the brain. So if this mechanism is at work in the brain, then the distinctive feature should be the emergence of coherent states. And interestingly or significantly, empirical evidence points now absolutely to the existence of this postulated mechanism. As I've already mentioned before, the neural correlates of consciousness indicate that conscious processes are characterized by the periodic formation of coherent states, with each coherent state representing a highly synchronous neural activity pattern. And this is known as gamma synchrony. And what you see here is basically the so-called theta cycle that takes place. So while I'm talking to you and while you are listening to me, we are conscious beings, and this theta cycle takes place in our brains. So every roughly 200 to 250 milliseconds, such a formation of a synchronous neural activity pattern takes place in our brains. And this is what the neuroscientists observe as the neural correlates of consciousness. But this process can now be understood at a deeper level as the periodic establishment of resonant brain zero-point field coupling, which leads to a sequence of modified zero-point field states, with each zero-point field state, each modified state being characterized by a set of amplified field modes. And it is this sequence of modified states in the universal substrate of consciousness that we perceive as a stream of consciousness. That would now be the deeper explanation of what is really going on while we are conscious. So let me summarize the talk and come to my conclusions. The presented approach to the understanding of consciousness is based on the notion that the omnipresent zero-point field is an inherently sentient medium. So we are not only, maybe we could put it this way, so far we have understood this omnipresent field only in physical terms, but now the key, maybe the key to the understanding of consciousness is to think of this field as a psychophysical entity. So the omnipresent field is not only this energy field, it's also an inherently sentient medium. That is now the essence. And in this understanding, the brain employs a universe. It does not invent consciousness from scratch. It does not produce consciousness from insentient matter. What the brain actually does, it employs a universal mechanism through which it taps into the phenomenal spectrum of the zero-point field. And this mechanism it's now very transparent. It's not magic. It can be described very clearly. It's resonant coupling of the zero-point field to the glutamate-water matrix. That is a basic mechanism that takes place in the brain. And this mechanism leads to the formation, propagation, and synchronization of coherence domains, and most importantly, to the amplification of zero-point field modes. And part one is important to understand the neural correlates of consciousness. Now we have a physical explanation, a very consistent physical description of how synchronized large-scale activity patterns are formed, namely by the formation, propagation, and synchronization of coherence domains. But the key to the understanding of consciousness lies in the zero-point field itself. So the crucial feature of the mechanism is now the amplification of selected modes of the zero-point field, which gives rise to a differentiated conscious state that emerges from the previously undifferentiated conscious background. This is the understanding that now emerges from this approach. And consequently, the formation of conscious states requires resonant brain zero-point field coupling, as this is the only form of organization that leads to the modification of the zero-point field. So the key feature of conscious systems is resonant interaction with the zero-point field, because this is the only form of organization. It's an organizational feature now. It's the only form of organization that leads to a modification of the field. All the other systems that do not play resonantly with the field cannot modify the field. And that's the reason why, for instance, classical systems cannot be conscious. They are unconscious because they cannot modify the field and cannot amplify selected field modes. And I think that this opens completely new vistas for our understanding of the fundamental mechanism underlying conscious processes. And that's what I do in my work, and driving this approach to a really self-consistent theory of consciousness is the part of my scientific work that I will pursue in the future. So thanks a lot for listening, and I'm waiting for feedback and for your questions. Thanks a lot.
Thank you, Joachim. This was really, really nice. I'm sorry that we're like on Zoom platform, so you cannot hear the applause, but I guess I think you can feel it, I hope. This was a very, very, very new, I've heard many new things that I was not aware of, and I have myself a lot of questions. But maybe since I'm the moderator, I should be nice and give other people a chance to ask questions. So we are ready. Joachim is ready. Who wants to ask first? You can raise hand. You can just start speaking. Well, since Mayumi-san. Dozo. Hi.
Yeah, thank you very much. Dr. Keppler, I'm so happy to hear your talk, because zero-point energy field is sort of, although I'm an educator, that is my home ground. And actually, okay. Yeah, and maybe I'm not sort of capable to ask you questions based on physics. However, I just wanted to let you know, in 1999, in San Francisco Bay Area, although that time was just a dawn of zero-point energy, physicists, conscious discussions and so on. We, including myself, co-launched Planetary Academic Federation, mainly based on field, zero-point energy field. And my part is how to apply that to education, meaning in everyday life, mostly intuitively, in alignment with zero-point energy. And I am so happy to hear the solid scientific, presentation, like experts like yourself, for me, just to listen to, being able to listen to your zero-point energy work, how it has been developed, including like a molecular level and also electromagnetic level. But in my understanding, that is much more, sensitive, much more higher or detailed or finite. Yeah, I do understand that. So I'm so happy. I'm so grateful. This is after 25 years work on this field, actually, in terms of applications. Thank you so much. With my heart, thank you very much. With my heart and appreciation to your work. Thank you very much.
Thank you, Mayumi. That was a very nice comment. Who else? Professor Vitiello?
Yes, okay.
Yes, just go ahead.
Yes. Yeah. Thank you, Joachim. Finally, we meet at least as electromagnetic images. Yeah.
Hi.
Yes. So thank you very much for your talk. It's very, very clear, and you see, I agree. I fully agree. Also because of course, I read your paper. So I could find myself, but there is one point which has to do with the communication problem. Please consider this point, communication problem. The zero-point field, which is there, I mean, but from a physicist point of view, it is always related with some material system. I mean, there is no abstract. This is a communication problem, because otherwise we risk to go to metaphysics, because we think something is out there pervading everything, and so is. But then there are dangers in the interpretations. of what you and also I agree myself, but not only myself, other people which are on this line. So for this reason, I was since long talking of the fact that living system, living matter in general, is an open system. As every other physical systems, we do not observe closed systems. So since these are, there is a dissipation, which means giving, but also receiving in both ways. But word is dissipation. Since dissipation is the key point, and you are using this aspect when you say that brain gets in some resonant mode with the zero-point field. So this dissipation requires, just because the mathematics we have is only for closed system. To all imperialistic, how to say, greetings and of mathematics, it is only for non-existing systems, the closed ones. So we need to work out with that mathematics how to deal with open systems. Now, since 1975, but even before by Araki, it was proposed to double the degrees of freedom. Namely, we have one system, which is an open system, think of a brain. Then, whatever it is, we have a copy of that brain, which is your 0-point, the resonant aspect of your 0-point field. So in this doubling of the degrees of freedom, we do have a formalism, which is one has been used in thermal field theory by Umezawa and Takahashi, and by myself in this study of the brain. Now, at this point, I would like to stress, I mean, these things are in your talk, so I'm not contradicting, I'm only rephrasing in some different way. Yeah. So at this point, the interaction of the system, the brain, if you want, with that part of the brain, which by weight, the water matrix is crucial. That is essential. It is a pity that biologists and neuroscientists, except a few, are just dismissing the water part of the story. But in any case, so that interaction comes together. And these two are even more than resonant. They are entangled. Because in the mathematics we work out, we can show that the state of the brain and the resonant 0-point field in our words, or my double in my words, just to make popular that idea, I call it my double, that part is an entangled part. Namely, one cannot exist without the other one. Now, I think that this conclusion, which is also your conclusion, which describes the consciousness, is crucial, because at this point, the consciousness has a basic material matrix, let's say, but is a functional activity. And this is why there are so many gaps in other approaches, because they do not consider this aspect. It is a functional activity. So this functional activity is being together today, for example, talking each other. Yeah, I wanted to stress and to how to say enlarge even more, if possible, your vision. Yeah, which I share. Yeah.
Absolutely. Thank you very much for the clarifications, for the additions, and I agree with you. Yeah.
Thank you, Professor Vitiello. Someone else? Antonella, please go ahead.
Thank you for your talk. Very interesting. I have a question. Which can be possible the effect of an external electromagnetic field on this configuration? In other words, which are the effects of electromagnetic field, the external electromagnetic field, on the structure that you have described. This is in general, and in particular, I saw years ago that the effect of a very weak electromagnetic field on glutamic acid is a huge effect, as the effect to deprotonize the glutamic acid, and then to pass from the configuration of the glutamate to the configuration of glutamic acid. I imagine that this could have a very strong effect on what you have described as the coherence of the column of the inside.
Yeah, I think so. I mean, I haven't, what you described, I think I have not done it mathematically so far, and just concentrated on, well, the 0-point field, on the normal, so to speak, the unperturbed interaction, that normally takes place in the brain. But I agree with you. Now, of course, when you apply external fields, you should see modifications particularly when you apply fields that have the right resonant frequency. Now you should be able to modify the interplay between, or this would affect the interplay between the microcolumns or the glutamate water matrix and the 0-point field. And yeah, I think that could result, for instance, in maybe also the breakup or the disruption of the interaction, or in other maybe also, but on the other hand also you can strengthen a certain couplings of certain microcolumns to the field. But maybe we should discuss separately. But I fully agree that the application of external fields that should be now should be describable in this formalism, and we should see changes in the coupling and changes in conscious states.
Thank you.
Thank you, Antonella. Professor Vitiello would like again to say something.
But if someone else want to put questions, I will reply after. I had on this specific argument something to say, but after someone else, if I do not want to monopolize.
Okay, someone else? Professor Miriam. Hello, Miriam. Very nice to see you. Please.
Nice to see you.
Go ahead.
Well, thank you very much, Professor Keppler. It was very interesting. And I understand this is theoretical physics. You said that the data agreed well with the microanatomy of the brain and the functional units, and I wonder if the application of this theory, when it is fully developed, will allow us to differentiate between conscious and unconscious people, or even to detect the level of consciousness in other animals. Is this being already experimented? Are there experiments regarding this approach?
Actually, one important part is now in the next step. I will formulate an experimental path, experimental strategies that derive from this approach. Now, one of the questions, of course, is how can we now convincingly show that the 0-point field is a carrier of consciousness? And that's what I'm currently working on. namely to specify a clever experiment to demonstrate the importance of the zero-point field for the formation of conscious states. And this would, of course, also apply to other animals, or to the brains of a rabbit, for instance, yeah, of other animals in the biosphere. And not only animals, because now we can think of this fundamental mechanism. And of course, one idea would be, well, in any system that shows, that displays this fundamental mechanism, we should expect the presence of consciousness. If the zero-point field is a carrier of consciousness, and if coupling to the zero-point field is the key mechanism for the formation of conscious states. And what I'm currently thinking about is to design a clever experiment that shows, that demonstrates the absence of phenomenal consciousness if we are able to disrupt the coupling of the brain to the zero-point field. So it's about finding a clever conditions, clever experiment, and a clever experimental setup that prevents resonant brain zero-point field coupling, and then shows that when we disrupt the coupling in certain areas of the brain, in certain microcolumns, then the phenomenal consciousness that is typically or normally associated with these microcolumns, then the conscious experience goes away, it vanishes. For instance, applying a certain setup, a certain shielding of cortical microcolumns from the zero-point field in a rabbit cortex, would prevent the rabbit from being conscious, and would result in a completely different behavior of the rabbit. So that would be now part of the strategy to show that this coupling is essential for the formation of conscious states. And when and once we have understood this and really verified, so to speak, this fundamental mechanism, we can draw conclusions about any system in the biosphere regarding the presence or absence of consciousness. That's my hope and my strategy for the future, to now derive clear empirical setups to show that this is not only a nice theory, but really a fundamental understanding of consciousness.
Thank you both, Professor Miriam, for this very interesting question. I had a similar question like yours, but maybe I'll have more time later to ask. Thank you, Joachim, for your answer. Someone else? There is one question in the chat box. Sorry. So it is from Nishiyama-san. Why don't you go on live? You can ask live. So the question is, if we construct artificial devices relevant with zero-point field in coherent domain, does that artificial device have consciousness? Yes, I also wanted to ask similar. If I put together glutamate and water, can I make somehow something artificially conscious?
Yeah, maybe also with other types of molecules. Evolution in the biosphere has obviously, or as we see, chosen glutamate as the most dominant resonant in combination with water, the glutamate-water matrix, as the resonator that interacts with the zero-point field. But of course, it could be also different types of molecules. Once we have understood the mechanism, we could also think about other types of molecules, not only glutamate, in artificial systems. And of course, one implication would be that putting highly concentrated, appropriate molecules in a container and letting them interact, or controlling their interaction with the zero-point field, would enable us to construct artificial conscious systems. And again, the key for consciousness would be this resonant interaction of the molecules with a zero-point field, which would also mean that today, present day computing devices are definitely not conscious. So if the theory is correct, then one implication would be, well, today's computing devices are not conscious. It's not a result of a calculation process. Any system that we want to transcend the barrier of consciousness needs to undergo a resonant interaction with a zero-point field. And only constructing units where we control concentrations of molecules that are then highly concentrated enough to interact resonantly with a zero-point field, only then will this unit exceed the threshold of consciousness and experience a phenomenal quality.
Very, very nice. Very nice said. I love how you absolutely insist about this, that the consciousness is a result of resonant interaction with zero-point field. So that is something that I could, that I could connect with my everyday life experience. When I suddenly understand something, or I suddenly find a solution for something, I cannot trace logically, I first thought this and then this. No, it just suddenly, appeared. I always like to say I'm a servant of my brain. It's not, I cannot guarantee if I can solve something or not. So it's just this so beautifully explains in a scientific way this phenomena that I'm just like, let's say, witnessing as a layman. Professor Tenkova, yes, please.
Joachim, thank you very much. I have few things. My first question was, why glutamate? I think I got some explanation, even also having Antonella's question. But I think that the concentration, we always tend to think about very high concentrations, but watermetrics is much better reorganized by low concentrations. And in water, we have different molecular structures that vibrate with different frequencies. So the glutamate concentrations might cause this difference and might be the source of these complex frequencies. I couldn't get very well. You talk about normal mode being a single frequency, and related to a single microcolumn, or having a pattern of frequencies that is related to different columns. Because to me, consciousness should be a spectral pattern based on different frequencies with different amplitudes, resonating with the 0 point. And what Giuseppe noticed about my double, I think that this huge number of frequency to be coherent with. So we could have coherence in water at different frequencies. And this is when we have the mirror frequencies in the 0 point. So most probably, why not having water and water? And producing different frequencies from this water glutamate ensemble by different concentrations of glutamate, as your hypothesis, maybe other molecules as well, resonating in the water in our double.
Yeah, as I said, I mean, water plays certainly a crucial role because it's kind of an amplifier in the whole game. But first of all, the mathematical analysis shows that you need to exceed a certain level of concentration. And water, yeah, is only highly concentrated, but you need to, maybe you're correct. It's the release of glutamate into the matrix. Maybe that's the crucial point. Yeah, that then triggers the coupling of the matrix to the 0-point field.
Yes.
But
Yes.
you need some triggers in the microcolumns. It cannot be water itself because pretty stable, and the water concentration is not regulated on this oscillatory level. Yeah, what you regulate is the neurotransmitter concentration. So at least partly, I would certainly agree with you that maybe it's this two-step process where the release of glutamate then triggers this coupling of the matrix to the 0-point field. Yeah.
Yeah.
So
Really
the reason
religious.
why glutamate, but glutamate could do the job. One important point is the following. You may think, well, there is only one frequency that plays a role. So this, and that's correct. There is one dominant frequency that drives the resonance between the glutamate molecules and the 0-point field. However, the calculations show that nevertheless, the amplification of the field mode is microcolumn specific, and that's why, that is because that is part of the calculation that can be shown that you have a shift in the resonance frequency that depends on the tissue concentration. So this would give now every single microcolumn the possibility, depending on the local tissue concentration, to really change
the frequency.
A different frequency. So,
Yes.
And that could explain why every microcolumn has its specific frequency.
Yes.
So you have a spectrum of frequencies
Yes.
all over the cortex.
You have a spectral pattern then.
Yeah.
Yes.
Yeah.
You have a spectral pattern that fits with the double.
Yeah.
I still think that this double is more.
Yes. Yeah, I haven't thought about that actually. Maybe we are and Giuseppe and I, we are much closer together than I thought.
You're right.
Video, go. Yeah, Giuseppe, go ahead, please.
Yeah, may I? If anybody wants, I have a few things to say about some questions too, because yours, for example. May I?
Yes, obviously.
Okay.
Just go ahead.
So let me start from Yelena. You said, and also someone, I missed the name, who was asking how to do artificial consciousness device. I wrote the device because it's not a machine if it has a consciousness. I answered to such a question in a conference, I remember, to a big shot on the Blue Project on Brain in US and Europe. Just make a baby. It will be conscious. It's much more easier and it works much better. Unless you want that it could be suicide. Then unless you want workers which cannot strike, or military soldiers which can die, and then you make money, not only because you conquer a new country, but also because you produce new of such devices. So one must be very careful. In any case, you might call it, if you will succeed in doing such a device, you must call it Spartacus. Because there is no reason why it should listen to you, which is your first plan. So Spartacus, make a baby, is much better. And then, okay, there was the question by Antonella De Nino. if the point is that she was asking about, just to remember, the point is that what is the action of an electromagnetic perturbance, let's say external field, how is working? Now, we did a computation with Emilio and Giuliano Preparata in 1988. The paper is on Physical Review Letters, where we were considering an external electromagnetic field, and we were generating coherent domains. These coherent domains were depending on the presence or not of that coherent, of that external field. If instead you do, how to say, an updated computation by including the electromagnetic field as a field, as a dynamical field in the Hamiltonian, then you get what was worked out by Emilio Del Giudice, I think Mele, and Giuliano Preparata. You get one more equation for the electromagnetic field, and that is has been also used by Joaquin in doing the computation. So what is the conclusion? The electromagnetic field may interfere with the coherence, which is a key point, because coherence means collective modes, which means possibility to tune with external 0-point field. I say 0-point field, you will say double, it's okay. So this is the action. Is a perturbation in the tuning of two systems. It will act. Now, one experimental, how to say, observation, of course, one can do what you plan to do, Joachim, but there is one very simple way to do it, anesthetics. When we are asleep, our consciousness is still there. Physicians, they have a full classification of coma of several kinds and so and so. But when we sleep, we dream. The dreaming is a moment in which we identify ourselves. We lose, I mean, the identification of ourselves, and we identify ourselves with our double. So we lose time ordering, and we lose logical ordering. So we see dreams, which are more or less gratuitous scenery. So that is a very good experiment from a living experiment, let's say. If we isolate a system, then consciousness goes down immediately. Put your cat asleep. The cat is a beautiful animal because it's very difficult for the cat to get asleep. In any case, so I think that this the presence of that external field has to do with that. Why glutamate? I think that the beautifulness of water is that water forgets oblivion. That is beautiful, because otherwise we would be crystals, and we will not be conscious within a range, because we have only a definite band of frequencies to match with external world. But we have to tune to other frequencies, to other coherent. So my hypothesis is that glutamate acts as a tuning knot for different tissues, different microcolumns, and so on. So in this sense, also to Rumiana's question, this might be. But all of it keeps together beautifully, I think. Thank you.
Just to add something to Giuseppe's, with anesthesia, we get less free water molecules, and we get lots of hydrogen bonded water structures. Exactly. So that's why, yes, that's why water is not acting as an engine, as I say. So it stays as a battery. So we don't have this active part of the water. And that explains why there's no communication with the field.
Yeah, and we might do an experiment how in sleeping people glutamate works to differentiate the two dynamical regimes in the same living system, awake and sleeping. The glutamate action, how it works in these two cases. I think when we sleep, I don't know, but I would like that when we sleep, glutamate is almost useless, just because of what you said, Rumiana, in the sense that water cannot make many coherent domains. It sticks on some ordered structure with more hydrogen bond, and that's it. I don't know.
Yeah, can I ask one more question to Joachim? Joachim, how about microtubules and tubulin in the whole picture? I was just thinking.
Is there, Joachim?
I wouldn't rule. I mean, I wouldn't rule them out as maybe important partial aspects of the whole dynamical process, but I personally think they do not play a major role or the key role regarding this interaction with the zero-point field. I think it's mainly triggered by glutamate, by the neurotransmitters, and of course, as we all agree, water plays a dominant role in this dynamical interaction, and in driving different modes or in selecting different modes, which are maybe then microcolumn specific. But I mean, it's for me, it's not. For me, the microtubules are not the control parameters of this dynamical interplay, and I would not attach the great deal of importance to them.
I was thinking that microcolumns might be related to microtubules. Microcolumns, each.
I mean, it's a scaffolding. It's a structural setup of the whole system. Yeah, that is somehow, of course, also stabilized and stabilized by the microtubules. But I mean, dynamically, I do not think that they are the key players in this dynamical interaction. But that's.
I don't think it excludes. I think both of the things are important, but for different reasons. I could agree. For example, it seems very logical for emergence of consciousness that glutamate water matrix is the most important, and maybe the nature for some reason favors glutamate because it perhaps guides this phase transition most precisely in order for the brain to enable this resonant interaction with 0.3. With microtubules, I think they have maybe, now when I listened to your lecture, I think they have actually another role, and that is in the forming of the memory. So it is another thing, storage of information than consciousness. It's a little bit different. It's like a little bit different role, isn't it?
I mean, I also would think that Giuseppe agrees with me. Following my approach consistently leads finally to the conclusion that there is no memory stored at all in the brain, but the brain is just the right readhead. It's just the right readhead. It's a translator, and all our autobiographical memory is, at the end of the day, it's stored, it's conserved in the zero-point field. And that's the storage of everything, the cosmic memory where we write and read, where we write into and read from, And all the rest in our brain is just the dynamical resonator that writes information states into the field and reads information states from the field.
But then it's like
but I think to really prove this, that's an even higher challenge.
Yeah, yes.
But Walter Freeman and Carl Pribram, they wrote both in different papers that they could slice the brain of small animals, and the brain was still remembering, slicing those parts of the brain where neuroscience thinks that are sites of memories. So this dynamical view of memorizing is true. You were talking of self-orchestration or orchestration, something a word which in more traditional terms are collective modes, namely coherent modes.
Yes.
Microtubula, they depend on the water polarization, because we did compute the inner diameter of microtubulus starting just from water, and we found about 15, it was 14.6, but it's 15 nanometers, which is what people observe. So water polarization is at the basis of the construction and keeping on microtubulus. Microtubulus, as Yelena was saying, maybe they have a different role and importance. Namely, we know that most of the metabolic activity goes along microtubula. So you must keep the system alive.
Sure.
So it's of course a non-linear system. It's not just A cause of B and so on. So you must keep the system alive. Water polarized, you have a microtubula, and the microtubula, you have a metabolic activity, and water may be continuously undergoing phase transitions, which was the title of a paper with Emilio Del Giudice and Richard Manca many years ago. So in this sense, microtubular is essential, but not, as Joachim was saying, so crucial in the specific task of the functional activity of consciousness. I agree fully with you. Yeah. So they are they came in the game like my hands. I mean, they came in my consciousness because otherwise I cannot touch things. Yeah.
Yeah, I think we need an ultimate laser in the whole story, and the water has to be able to produce all the frequencies all over. So we need a mechanism for this.
Yeah, and maybe glutamate has such a role. You dope water, so you tune the range of frequency which water can accommodate. Maybe.
Yeah. That's I'm close. I think our way of thinking is very similar. The doping of water and the resonant interactions then with a water container that takes place via the zero-point field.
Yeah, water as a structure could be a sense. It is the ultimate sensor, and it is the ultimate light source, a coherent light source. So, and we need coherent light at all possible frequencies, and that is what water provides. And then the laser and the sensor makes the feedback system.
May I say something about? I agree. Can you imagine how much I agree on water? But we have a problem to understand how a feeling. If I'm happy, like in the picture, Joachim, he did show us of him on the rocks sitting. He was happy at that moment. How happiness and also painful experience, how such kind of emotional, how it is connected to biochemist.
Beautifully.
that is the crucial point, because now the connection is through the coherence of the water, because coherence is tuning the orchestration. Let me use Joachim words. The orchestration of the system. In this case, of microcolumns. The orchestration of, and that orchestration is the key to through microtubula to metabolic activity. We have experiments in which microtubula disassemble, or they became more, the net of the cytoskeleton becomes more vigorous, more active, according to sounds, just words or music. And this is has been done in Padova by some colleague. We wrote a paper together. Who they are cardiosurgeon. So the cell was a HL1 cell from the heart, and it was subjected to noise or to mantra or to words. And they observed how microtubula, they were disassembling or reassembling, or the cell was dying in some stressful situation. So such a kind of very light, I'm thinking.
Low energy, yes.
Yes, I'm thinking now of Antonella question. Very light effects, phonons, in this case, words or music, may affect biochemists.
Yes, we published a paper on the influence of the frequencies on the water structure.
On this kind of system.
Yes. Yes.
It was not so okay. They the people that did the experiment, they were physicians and biologists, so they did not care much about frequency ranges, which was my complaint. But in any case, there was the effect. There was the effect, and now I propose to do it again by looking at the specific frequencies. Yeah, and but that is crucial point. So this is why it's not just water; it's also biochemist. It is also biochemist.
No. But at the end, it's always water, I think.
No. Okay. But why? Give a room also to them, otherwise they will kill you. They do not know why a clown is accepted in the hospital. Why a physician in a certain situation tells you must be strong with your mind to face such a pathology. What that means? There is no chemist in there. No, there is the chemist, because such feelings through to water, they act on biochemists, like meridians in acupuncture. They operate at a physical level, but they have a chemical effect. This is crucial to understand. In fact, we did, I mean, I have another paper with Patrizia and Enrico on meridians, connecting such two sides. Biochemistry is there. We should not just think of water is 90% of molecules is water. Of course, it's there.
Yes.
But if the water is the phase, the connection with the consciousness, that this is what makes the connection between chemistry and physical.
Maybe all these elements have their own frequencies, so they are also fine-tuning specific frequencies.
Yes.
This is what I'm thinking, for example, why certain chemicals produce certain reaction.
Yeah, this is why I like chocolate.
Exactly. That was my point. I was listening to you, and and I was thinking about, and I remember once, for example, when I was younger, I was ill, me and my sister, and we got the same drug from the doctor, and she was well after the drug, while I literally saw dragons, like, and I was fascinated. How come, like, that was the same substance and such a drastic, like, how can I say, different picture of reality, So that was, for me, like, I could not believe that these tiny molecules can shape the way I perceive the world. So I agree. I mean, water is absolutely, I think, matrix where all these things happen. But I think this some kind of fine tuning is achieved by, otherwise we wouldn't have these different neurotransmitters,
Absolutely.
right?
Yes,
And
yes.
so they have different colors, let's say, to our perception, shades of consciousness, it seems.
That's my point of view. Yeah.
Yeah, I understood, and I really, really loved how you put it. I love to have,
Beautiful.
like, scientific explanation for all these things that like, I always wondered how come, this happened, and so on. Anyone else would like to... Miriam, yes, of course.
Miriam?
Go ahead.
Hello. Perhaps I don't know if you find it related, but in a cell biologist point of view, a cell is only alive when there is an electrochemical balance or field across the membrane. I mean, and perhaps that is the connection between physics and biochemistry. Cells respond to electric signals through chemical shift of ionic charges, and that's why neurotransmitters also excitate or inhibit cells through opening or closing ionic channels in the membrane, and then increasing or decreasing electric potentials. So for us, for a biochemist or cell biologist, the way to learn if a cell is alive or dead is to observe if this potential, this membrane potential, exists or not, and perhaps connects everything: water, electromagnetic fields, and biochemistry.
Yeah.
That's, by the way, that's part of also the calculations I did. When you do the calculations, you find that the resonant interaction, let's say, of the glutamate water matrix with a 0-point field induces a kind of oscillating field in the microcolumns, and that would, in your terms, also apply to cells, to singular cells. And this oscillating intracolumnar field then triggers membrane potentials and the ion flows, which lead to the external outputs of the microcolumn. So this going along the lines of this picture, this all seems to play together now. Yeah? So it's this resonant interaction that leads to a intracolumnar oscillating electromagnetic field that then drives ion flows that lead to external outputs, and so we have the propagation of coherence domains, and so that's part of the picture that now sets in, and that confirms what you Miriam, what you was just explaining to us, I think.
And those oscillations, are they with specific frequencies for different columns?
Yeah,
Okay.
absolutely. So it's a shift, yeah. You can.
Yes.
So
Yes.
the main dominant resonant frequency is in the terahertz region of the glutamate water matrix with a 0-point field according to the concentration level, and so that you encounter in the microcolumns, and the calculations show that when this balanced state, the formation of a coherent, the stationary state establishes, you have a shift to the microwave region to 30 gigahertz, and this is known as a frequency band that has a huge impact, a huge influence on the triggering of axonal spikes. So you can do experiments, and you find that in the microwave frequency range, you can trigger or affect the axonal outputs of neurons, and so that gives now a nice consistent picture. I think that it is it's this bottom-up this bottom-up interaction with a 0-point field that leads to the formation of coherence domains and the propagation of coherence domains, and all this now seems to be part of a consistent picture you get. And also the size of the microcolumns. That's now a self-stabilizing system. So it's when you calculate the interaction, the evolution equations describing the dynamical interaction between the 0-point field and the glutamate water matrix, show you that the size, the diameter of a coherence domain is 30 micrometers, and that's just exactly the size of a microcolumn. So that seems to be a self-organizing process in the brain that leads to the formation of microcolumns, because the size is exactly the size of a coherence domain that is established due to the resonant interaction with the 0-point field.
May I add something on this point?
Mm-hmm.
May I?
Sure.
To Miriam. Exactly what you said is exactly what in some very general way in physics you say that the cell is an open system. Herbert Fröhlich, he writes that in 1938 he was astonished that the potential difference on the membrane of a cell is about now something like, if I'm not wrong, 100 millivolts, which corresponds, due to the thickness of the membrane, to fields of 10 to 15 volts per meter, which is breaking any material that we know, at least the functional properties of any dielectrics. He was an expert, one of the chief in dielectric materials. So the point is that once you realize that you have such a kind of activity, and this activity is based on ion flux, on biochemical reaction, then there is something left to understand. How randomness, which is the key point of any chemical reaction, how randomness may produce, as Schrödinger was saying, regularity not in average, but highly efficient systems. And so this is the point. That point is perhaps understood if one considers that the physical dynamics underlying the chemist, the biochemical activity, is the one of coherence or orchestration, as Joachim said.
Mm-hmm.
So that is what is stemming at very low probabilities that the two molecules cross together and make a reaction. Otherwise, you cannot understand how this happens. I always make an example like this one. We are talking, each one of us, I mean, I especially, sorry, but which one is the probability, Miriam, that we met on a Monday morning for us in Europe, at least, talking about these things among how many billions of people we are on the planet? Billions, okay? Because we share the same interest in something. So the probability that we met, which would be otherwise zero, is now not zero. In fact, we are here since more than 1 hour. So this is the physical basis of what Joachim was talking, and there is a dynamics behind the biochemist, which produce highly efficient systems.
Beautifully said. We have one question actually from Wieland Lackinger. Go ahead. Thank you for joining us.
Yeah, thanks for organizing this kind of meetings. It's very interesting for me, even when I'm a master student. So the question of Miriam reminded me at the experiments of Mr. Levin with his worms. So my question to Mr. Keppler, do you know these experiments? And if yes, is there a connection to your theory?
No, unfortunately, I do not. I don't remember at least, but I think I do not know the experiments so far.
Okay,
no.
So then, yeah, sorry.
No, not sorry, but maybe I should be aware, should get aware of these experiments and read something about these experiments after the session. Yeah.
Yeah, they are very, very interesting. He cuts the brain of worms, and then he put an electrochemical gradient on it, and then they remember the sources of food after their head grows again. And so my question was where the information is saved when not in the brain. So maybe there's a connection to your theory.
Yeah, okay. And who was the one who performed the experiments?
His name is Levin.
Levin.
Levin or Levin. I don't know how to pronounce it.
Interesting experiments.
I'll look it up. I'll look it up.
In German,
luckily it's worms.
Are, yeah, yeah. In German, the word is Flatwurm. So
flatworm. Yeah, I also wanted, it is a little bit related to what you just asked, Wieland. So the entire story from today's lecture, I understood it is actually just related to this neocortex. So everything is about cortex.
Yeah, but in subcortical areas, I think very similar processes take place. So it would be, I think, not correct to assume that only the cortex is able to make this dynamical interaction. But as far as our everyday conscious experiences are concerned, of course, the neocortex plays a dominant role. And that's why my analysis and my work is puts special emphasis on the structure of the cortex and on the role of the microcolumns in this dynamical interplay. That's the main reason to have. When you do a rigorous mathematical analysis, you have to start somewhere, and a natural point of departure for me was starting with the microcolumn. And that gives, as I said and explained, a very coherent picture, but it does not exclude other parts of the brain or even other parts of other animals to be to play a role in conscious processes.
Yes, absolutely. We are approaching, we should be finishing our webinar. Do we have more questions from the audience? If not, I actually wanted to ask you, Joachim, one question even before we began the webinar. I'm curious, what made you, what was the, I mean, do you have some kind of, let's say, a memory or event? What happened that you decided that this is going to be what you want to really research? I'm really curious about that.
Yeah, but I mean, it's really, yeah, it's thrilling for me to get a definite answer to the question of what differentiates a conscious system from a non-conscious system, and that question has it was from early childhood on that I asked this question, and I never got convincing answers from my parents or from anybody else in my environment. And I thought, well, I must. It's worth studying this topic because it must be so. You cannot give an easy answer to it. Yeah, so one should study it in more detail, and that's the reason why it became my passion in life.
Ah, I see. Yes. So you obviously resonated.
It actually started many years ago, and the reason why the typical, the mainstream answers never convinced me. That's the reason why I started to dig deeper and to do my own research on it.
Thank you very much for answering. I really wanted to ask you this. Anyone else? Anyone else? Anything to comment or add? Okay, we had some comments on the chat box. Actually, some people had to leave, but I could perhaps read them. Okay, this is from Igor Yerman. He provided one link for the paper on this brain regeneration thing that Wieland was mentioning.
Okay.
And from Fiorenzo Marinelli, unfortunately, he was happy to be here, and he would like to hear this discussion again. So of course, this webinar is being recorded, and it will be in few days available on YouTube, and everyone will receive a link. And of course, when it is on YouTube, you can add your comment.
Jelena, we don't hear you. No. Okay. Meanwhile. Okay.
In the meanwhile.
Works.
Do you hear me? Do we hear me?
Yes.
Unfortunately, new headphones. Okay. So I just wanted to say, in a few days there will be, of course, recorded version of the webinar on YouTube. So whoever has questions, comments, they can also place them there below the video, and Joachim can also see and even like answer there if he can. There is one question from Edward. Hello, Edward. Aime. Have we established an interrelationship at the cellular level between sound, light, and the scalar scale and their activity at the cerebral level? Interesting.
Not yet, I fear. I'm just in the process of it's going to be a longer journey, I think. It started with a philosophical approach, and then it got into neuroscience, and now I think we have, at least from my point of view, a very clear mathematical, physical picture of what's going on in the brain, and the journey will hopefully lead us to more and more advanced and deeper understanding of these resonant processes, but
most probably,
moment, I cannot answer all the questions.
Yeah, most probably it will lead you to spectroscopy.
Yeah.
I think Joachim definitely needs help. I mean, he cannot do this all alone, and we all have so welcome, many.
Yeah.
there is
frequency.
one comment from Shima-san, from Swiss Alternative Medicine in Geneva. Maybe the similar experience can be experiment, I guess, can be conducted on a hippocampus where the memory is stored as a classical storage part of the brain. I really love that you said this, because when I asked if this is only related for neocortex, I was also wondering how the hippocampus and these evolutionary older parts of brain are involved, like amygdala.
They're all involved.
They're all.
Amygdala, certainly the thalamus. The thalamus plays an enormously important role in these thalamocortical loops, and so it's a driver of cortical activity. Actually, everything goes through the thalamus, and the hippocampus is also involved. That's the reason why the key message is, well, in understanding our somatosensory, tactile, olfactory, visual experience, most of it is connected to the neocortex. That's the reason why, of course, you study it, and I started it with this part of the brain, but for sure, all the other parts are also involved when it comes to building up this dynamical interaction with the 0-point field. And that's right. Also, the hippocampus plays an important role. And by the way, the hippocampus is involved in this theta cycle that I was mentioning before, this oscillatory, this regular establishment of large-scale synchronized activity patterns. Also, the hippocampus is involved in the theta cycle, and that's the reason why, of course, also the hippocampus should be involved in this resonant interaction.
Great. Wow. And it colors, I mean, I know that it is involved in how what do we actually perceive. It colors our perception, the involvement of hippocampus. I think we have extended again this webinar for much longer than we're supposed to. If there is one more short question, I think we have some time. Okay, we have one more comment, and I will read this comment, and I guess then after that we can proceed to say goodbyes. So from Eduard, it has been established that our microbiota is not the second. Oh God, yes, I'm also interested in this brain gut. Oh my God. But it has been established that 20% of data comes from the brain and 80% from the microbiota, and that the vagus nerve is our information superhighway connecting them. Is there a relationship? The things are becoming very complex.
Of course, you have the input channels to the brain, but I think most of it, what I was explaining to you, most of the processes that I'm studying that do take place in the brain. Even so, of course, there are information highways that induce information into the system.
Yes.
Yeah. And this is something that actually reminds me very much of one great woman who is no longer with us, unfortunately, Mei Wan Ho, who once said something, whenever we say I or me, just remember, like, for example, in our human body, at least 2 kilos of our weight are these pathogens or bacteria, viruses. And whenever you say I or me, they say me too, me too. So there is no really closed system, as Professor Vitiello is saying. We're always in some kind of symbiosis, and we're always, I guess, connected to that field. So it's never just me. We're not. No man is an island.
Yeah.
Well, okay. Unfortunately, it is really like 2 hours. I'm very, very happy, and I so enjoyed this webinar. We have comments from Shima. Shima, thank you so much for asking and commenting on this. It is wonderful to see you here. Joachim, thank you so much for this very inspiring lecture. I heard many, many new things, and I think other people as well. So as I said, in a few days, this will be available on YouTube, where you can comment, share your like questions, answers, and so on. And of course, on YouTube, you can have you will have available translation in Japanese, especially. This is fantastic for our Japanese audience. And I guess this would be it for today. Thank you all who asked questions, who attended today. It was really beautiful. Thank you, Professor Vitiello, for contributing so much to this beautiful discussion. I always enjoy hearing you speak.
I would like to, yeah, Jelena.
Professor Senkova, yes.
Yeah, just thank you all. I think we had a wonderful discussion. Joachim, very consistent and wonderful story and material for discussion. And I would like to keep this webinar in this with this atmosphere. So we learn a lot from each other, and we could discuss. And let's continue. I think we'll go somewhere in a very, we'll get there.
And we should work more, I guess. Joachim cannot do all this by himself. We should try learning more physics, help with the equations.
As I said, support is welcome, and I would like to thank you, all of you, for the discussion, for the valuable comments, and for having the opportunity to talk to you. Many thanks again for the invitation.
Thank you.
You're very, very welcome. Well, this would be it for today. Thank you all again, and enjoy your day, Europe. Good night, Japan. Have sweet dreams. And I guess see you soon then. Professor Vitiello.
Okay, bye-bye. Thanks a lot.
Vitiello, I will have to write to you about something that you said at a previous webinar. Actually, you said it today also about how the beauty of water is that it forgets. The oblivion of water. For me, that was so beautiful when I was sharing the information about your video on YouTube. I had to cite that. I quoted that on all my social networks. And then when I was recently at a beautiful conference in Geneva, I saw one of the speakers actually shared exactly the same quote from your lecture. I was really fascinated. I was so happy to see that. I love that. I think it will be something like part of the history, what you said about the phase transition and how it is important for life.
Yes.
Thank you very much.
Livio, hello. Thank you very much for coming.
Thank you.
Professor Giuliani. Professor Giuliani, see you in February.
Yes. Looking forward to that. Bye-bye.
Thank you all. Thank you very much.
Thank you,
Bye-bye.
Thank you very much. Thank you,
Bye,
Jelena.
Eduardo. Bye. Bye, Professor.
Thank you, Rumiana.
Thank you.
Okay. 3, 2, 1.
Bye.